Okay, good morning. Thanks for everybody who are here. And so this is the Pharmaceutical Science Committee and the Drug Information Subcommittee joint meeting. So this is the first time I run the hybrid and in-person meeting concurrently, so just bear with me if I mess up somewhere. Okay, so this is part of the agenda. So we will start with an administrative report from the Pharmaceutical Science Committee, and then the Drug Information Subcommittee, which is chaired by Andrew Smith, will be next to give everybody the update. And then we have a special presentation today by Dan Hurst. And then there's some update also. We may or may not have some update, depends on the participation of people virtual as well. But the main discussion will be in the first half of that. So first of all, we're going to just start with the committee administrative report. So you guys may remember the last 2 years we've been doing virtual, and I have been running the meeting virtual also. So the meeting minutes, actually the last meeting, I didn't realize there was no written report, but actually you can listen to a YouTube video. So that is the link for the YouTube video that recorded the virtual meeting. And then we're going to do a review of the committee membership, which I typically receive prior to each of the group meeting. There will be introduction of new members, etc. And then we have some discussion of some recent development in the area of APP, as well as the rollout of the Aurora from NCI for IDS management. Oh, sorry, wrong button. See, all right. I'm used to the up down button. Okay, so apologize for the quality, but that's the best that I can do based on what I receive. It looks like a membership list from about 50 years ago. So basically, usually we ask members to look through the membership list to make sure that we have members that we did not miss, as well as members that hasn't been here for a while, and then we'll talk about whether we want to retain them or not. We did have a few new members joined last year. Very happy about that. And in addition to that, we also 2 years ago started this program to bring in resident pharmacists from University of Michigan and Johns Hopkins as supporting us to write Section 3 of the protocol, and so that our member who are full-time working people will serve the role as the reviewer, and it helps to enhance the educational opportunities for the resident pharmacists, as well as to relieve the full-time pharmacists, working pharmacists of some time, so that is better manage the time also. So consequence to that, I'm very proud to say that the inaugural two resident pharmacists who graduated with the first group that work with us. Both of them are members, and one of them is sitting next to me, and he is now the chair of the Drug Information Subcommittee. And then I also have Michelle. She is at Wisconsin, and even she's not technically at a SWOG institution, she have asked to stay on as a member. So I am very excited and pleased about that. So now we have more trainees working with us. So we have 4 trainees, actually 3 trainees working with us. So we hope that they will also, as they finish, they will continue their membership with us also. And I also have one member, new member, application pending at this point from Virginia. So what I'm going to do is I will send out this list electronically, and then perhaps the members can take a look at that and give me their advice on the current membership roster. So introduction of new PSC member, actually, I already introduced the first 3, and they're all oncology clinical specialists from University of Michigan. And thank you for Andrew's recruitment process. But when we introduced them last time, the membership was not finalized. So I'm proud to say that all 3 memberships are now finalized, and they're also placed on various disease committee as liaison pharmacists also representing the committee. So we're very thrilled to see that even now stronger representation in each of the disease committee. And then the last person, Safis, I probably should not even attempt, is the membership application review pending, and she's from Fairfax, Virginia. So I look forward to hear the finalization of her membership application. And then we can start the process of placing her as a disease committee liaison pharmacist also. All right, so then is there any drug interaction initiative update that you would like to provide? If you can speak at the mic, so that they can capture.
Yeah, I'm not going to go back to the entire history. So sorry for those of you who don't know what we're talking about. For those of you who do, so we had the prospective study up and running, but it was having sort of trouble enrolling because of the strategy that we had used to actually run it, which was sort of through the ORPs as opposed to within a protocol. So at some point we decided that just wasn't going to work. So a year or so ago, we decided to discontinue that attempt at a study. There has been recent discussion within the Digital Engagement Committee about a project being developed by Dr. Nagla Karim, I believe. And her project is looking at using smart forms in particular to help with patient screening for clinical trials. So we had talked to her about the drug interaction thing and sort of thought about a combined protocol in using digital tools and technology to enhance sort of clinical trial workflow. So that is being developed within digital engagement in collaboration with the Thoracic Committee. And I believe that we have identified a protocol in the Thoracic Committee that is in development, that we will be integrating the drug interaction tool into the actual protocol itself. So Drug Interaction Initiative is moving forward within the context of one specific clinical trial, as opposed to across all clinical trials, which was the former process. We're just in sort of preliminary stages of discussing what that's going to look like, but at least the DE committee is behind it, and we have a medical oncologist clinical champion. So I think that there will be an opportunity to move forward, assuming the company is still on board. So that's where we are.
Is there interest in including more studies if there are additional suitable studies available?
I think trying to do it across multiple studies is part of the complication. And I think if we choose one study that has enough patients and has enough drug interaction concerns that we can test it within that one study at multiple centers, we'll get the pilot information that we need. So I think rather than complicate it by adding more studies, just finding that one good study and making sure that the Thoracic Committee is behind it is the way forward.
That sounds good. And Joyce, you're on the thoracic, right? The lung committee. Yeah. So maybe you can also kind of bring Joyce in to support whatever it is, because she's the disease liaison.
Yeah, absolutely.
Yeah. So she may be able to support in some way also. So we're going to stay with one protocol for now. Okay.
Yeah, and it's actually going to be embedded into the protocol itself.
Good.
And there will either be some kind of pre-post or randomized by sites or some kind of process where we can make some kind of comparison, but in the context of one clinical trial where it's part of the actual protocol.
Has any of the implementation of the APP thing being part of the discussion, because now there may be more people involved in ordering and things like that? I don't know if you. I'm
not familiar with what you're referring to.
So probably Joyce will be updating us, quickly on the agenda, but maybe something to also consider because with the implementation of the APP, there may be more people involved that can support and drive for that also. But we'll have Joyce give us an update and then we can think about that as well. Thank you. Make sure to get the right device. Okay. Huh? Okay. Is any questions or any comments regarding that from the audience? Okay, so we'll move on to the, too fast. The next one is, some of you may know that NCI is rolling out a thing called Advanced Practice Provider. And so Joyce Lee, who's our pharmacist liaison on the lung committee and also from UC Davis. And so I asked her to kind of work with the working group in be more active, engaged in all this implementation process. So she gave a presentation at the training symposium at this meeting 2 days ago. So Joyce, would you like to perhaps, in addition to describing your experience with the training symposium, perhaps give people a little background about that? Thank you. Yes.
Yeah, hi everyone. I'm Joyce from UC Davis. So yeah, I have the honor to give a little short presentation at the APP symposium on Wednesday. It's a 3-hour workshop for the APP or anybody who interested in the topic. The goal is to enhance the APP's involvement in research. So I give a presentation regarding just protocol review about those modification and those calculations. I think it was a really great experience in collaboration between the nurse research committee and palliative end of life committee, and those are the sponsor of this symposium. So I think that within pharmacy, there are a lot of APP out there, and I know that there are limited states that recognize this status, the APP, but I think there are a lot of opportunity for pharmacists involvement in research, like what Dan is doing, a lot of translational research and other perhaps like supportive medications or supportive care type of research. I think that we can definitely bring it back to our committee and think about whoever is already practices at APP, or try to encourage our colleagues to be more involved in the research process. Is there anything else? I think that's pretty much what I want to share. Oh, yeah. And then maybe the NCI, I don't know if that is anything. So I think with the related topic is the NCI also has recently created that status of the non-physician, I'm sorry, Matt, I think, yeah, the NPI, yeah, non-physician practitioner category. So I think that is another sort of platform for pharmacists as a practitioner in the oncology area. So I think that's.
I think from the pharmacy side, it's a little bit more because as some of you know, in 7 states in this country, there's already the state regulatory having this advanced practice provider, right? That's the term. Yeah, it's called APP also, it's so confusing. Yeah. But yet all of them has different guidelines of the scope of what that APP can do in each of the state. But nationally, that's not uniform. I think a lot of states are trying to get that status through the state legislation also. So consequence to that, I think the alignment of that is somewhat challenging, right? Because in the language with the APP, you also have to comply with the state regulatory stuff as well as your institutional. So people really need to be looking at all three of them to look for that alignment before they can be actively involved. So I think from the pharmacy perspective, in some way it's good that we already have the advanced provider in limited states, but at the same time, it makes it a little bit harder to implement because of this. It's not consistent across the whole nation also. So, but I think everybody is still excited about that. So we think this is going to make research and enrollment better. So, but it's definitely an interprofessional topic, and we will continue to engage in that. And there's going to be a working group form, and it's going to be interprofessional. So it will allows everybody to voice and help to continue to get this program moving towards maturation also. So.
And maybe I just want to share too, during that workshop, they do, as mentioned, as Silver mentioned, they're looking to create a work group for APP. So there's a link, and I can share, I can provide to you and share with the member. They're looking for interest, and so whoever want to involve in this work group, you can submit your application, and they have they have a call for it, basically. Yes,
Great.
thank you.
Thanks. Matt, do you have anything to add for the APP program?
Well, as far as the NCI is concerned, it was probably about a year ago we did.
Oh, can you go up to the?
Sure. So about a year ago, we did implement the policy across the NCI trial system that non-physician investigators, NPIVRs, if you're registered at that level within RCR, that you can write for IND agents, you can write for standard of care, which was previously prohibited. So it's kind of opened up that avenue for pharmacists, nurses, advanced practice folks that register at that level, that they can do that. And they have to be listed on the delegation of tasks log. That's an option whenever all of the protocols have it pretty much now, but they have to be listed on there.
That's good, because then that will lessen by protocol violation in audits, right? For people like, because in institution a lot of time there's a lot of verbal order being written, right?
Yeah, and previously those had to be co-signed by a registered investigator. That's no longer the case with this new policy. So it frees up some investigator time, and it gives some other authority to the advanced practice providers.
So my question for that is, but the pharmacist will still need to have the APP status within the state, right?
Sure, it's all
Yeah.
governed by whatever the state and local regulations. So obviously we can tell you you can do it, but it's, if your institution doesn't allow that, then obviously you can't do it. And if your state doesn't do it, obviously you can't do that.
But that will be for not necessarily verbal orders, but just that provider can actually write the order independently, right? What you're mentioning, Joyce?
Yeah.
If they are provider status, they can independently write the order with that. But there are also other pharmacists who are not necessarily in the state with the advanced provider, but a lot of time you see people write verbal order, doctor, right? So, and usually that come back with a co-signature by the physician later. But I guess my question will be, if that is the case, for the NCI, would that be allowed?
Can they take a verbal order and have it co-signed?
And later, yeah,
Yeah, as long as it's co-signed by the investigator. If it's not,
okay,
if that's the normal policy for the hospital, for the state,
yeah,
if they allow a verbal order and then it gets co-signed by an investigator, that's fine. I mean, in general, we don't like verbal orders, but if you have a verbal order that's co-signed by an investigator, that's fine.
okay.
So I think that the question or the scenario that I can maybe give to you is, oftentimes the physician, signed like a set of orders, and there's some error, whatever. So the pharmacist has to call the physician to clarify the order and usually in the daily base, usually we'll usually take a verbal order because, the location or the doctor's in clinic. In the past, and that's what we were told by the auditor, that because of the CTAP rule, pharmacists actually not allowed to or not supposed to do without the co-signature. So is it true, or is it something that you can have a suggestion for us to do in order to sort of work in a daily basis? Because otherwise, sometimes it's challenging to find a physician to co-sign those clarification order, not really to initiate a new order, but like to clarify the existing orders that has some discrepancy.
Yes, so they told you that you could not have a verbal order executed without the co-signature? So I would have to see the situation to see that, but in general, I would have to see the situation before commenting officially.
Yeah, this is okay. So I think that's kind of the
Right.
maybe not, we need more clarification on that. I think that was the
Yeah.
part that is actually most challenging for us.
Right.
It's getting that.
I mean, that's part of the purpose of us oftentimes bringing up topics such as this for discussion, is to allow people in the real time to have a platform to share some of the experience that they're not sure, right? And so
Okay.
and we have a liaison person here, so that's like the perfect. And if there's anything that needs to be clarified now, this is an opportunity to bring it back for the clarification to occur. Sometimes things like that occur, is just a little misunderstanding at one point of, yeah.
And I'm sorry, one more question for kind of, hopefully I can get some clarification is the CTAP provider signing order, is it only applied to NCI drug, like, or any treatment within that protocol?
Yeah, it's permissible for the NPIVR to sign anything that they're authorized to as an NPIVR for treatment within the protocol. So it previously was restricted to like supportive care meds, but with the new policy, it's expanded to the IND agency and the remaining standard of care chemotherapy.
Thank you.
So I also want to acknowledge we have like 20-some people attending virtual. So for those of you, you also have an opportunity to ask questions. So if you need to do so, just raise your hand. Yes, we can see that, but you do have to push a button. And then I will call on you, and then we'll give you an opportunity to ask question. If we are not able to do that, just put it in the chat also, and then I can just read the message out, and then we can communicate through that route as well. So, and thank you. So hopefully we get some clarification after this meeting in the issues that was brought up. So the next topic is on Aurora. So this is an NCI electronic IDS management platform. So we learn about this, and learning the rollout time also now is intended for January 2023 for the phase 1. So the phase 1 is primarily just a drug ordering mechanism of the program, but there's another portion of the program that will be rolled out, which is more has to do with the DARF management also. So the phase 1 will be rolled out in beginning of 2023. So we had some discussion about that, and my understanding is, Matt, maybe you can just give a quick summary of that portion of it. And sorry, the date has changed already since I put the slides together.
Hot off the press.
Yeah, hot off the press.
So you're just looking for a description of the phase 1 portion? So OAOP is currently the method for ordering investigational agents from CTAP, as well as accessing IBs and stock recovery letters and things like that. That OAOP system is being replaced by Aurora, and you'll be able to do all of the same things and access the same things. It's just a different technology platform that we're using. Some of the old technology is being phased out and is no longer able to be updated. So we had to move forward with that first, And then once that gets a little bit settled, we're going to transition into the EDARF.
Okay. So, I mean, it seems like it's the modification or impact on the sites are minimal. Is there anything else that we need to alert the sites to be able to, as the system rolls out, any new registration, et cetera?
So if you have people that are part of your primary shipping designee package and can currently get into the system and order agents, then all of that authority will be transitioned with OAOP into Aurora from OAOP. So that should not be an issue.
Okay.
One thing that, well, a couple things with the rollout of the new system, we're going to allow multiple shipping addresses for a particular investigator. So if you've had to order for one investigator to get it to another location and order for that investigator to get it to your home location, you won't have to do that anymore, as long as their RCR profiles are updated to allow that.
Okay.
And then the second big thing is with the blinded trials, because of we're going to have a link directly with Open, it's not going to quite be real time, but it's going to be close. It will allow sites to order drug for that first shipment, which we don't do now. We wait till the following day or the day after to get that information from the cooperative group ops office with the randomization, but that's going to come with that link to Open. So sites are going to have to order their own first supply. They're used to ordering the subsequent supplies, but the first supply is going to have to be ordered through through Aurora.
Okay, so I just want to pause and give an opportunity if anybody here as well as virtual has any questions or concerns, since we have Matt here, so we can get questions, answers, etc. Okay, I don't see any reactions nor chat. So Matt, would you like to just briefly touch on the phase 2 when the anticipated rollout date and just a brief overview of what this will do?
Sure. So Aurora is a complete inventory management system from our perspective. That view isn't quite shared across all the sites. Based on some of the commercial off-the-shelf systems that are out there. But it was designed for the purposes of IND agent management from a sponsor perspective from the NCI. So the DARF, the electronic DARF, will probably be released about 2 months after. So probably sometime in March or April. And then within 6 months, or 6 months, not within 6 months, but in about 6 months, we would make that a required thing to transition to the EDARF. That's the kind of working timeline that we have now, obviously subject to change, but it won't be shorter, but it might be longer if we're doing implementation and depending on feedback we get from the sites.
So Aurora, are the sites supposed to only use it for CTAP drugs?
At this time, it is limited to CTAP IND agents. In the future, that was one of the things that we plan to open it up for non-CTAP IND agents, but that again, that's a TBD date.
Okay. So essentially, because a lot of sites are using commercial vendor also, so they would continue to use the commercial vendor for the non-CTAP drug for even management and things like that. Right?
Yeah, they would continue with whatever the protocol-based instructions are for accountability. If that's a paper system or if there's a different system that's required for the company-sponsored trials, then that would be.
Okay. But eventually, there is room that potentially Aurora would allow the non-CTAP drug to also use that system.
Correct. I mean, the system's been built to do that, to expand at a later time. But at this time, we're focusing on the IND agents out of CTAP. With the exception of LungMap, since we're distributing for LungMap, even though it's a SWOG study, that will be within the Aurora.
Okay. Again, I want to open up the floor for any questions from the attendees. There is a question on the chat. Are there any plans to integrate Aurora with currently utilized commercial system just to avoid duplication for sites effort?
I feel like that was a planted question.
Yeah, I'm making up. That was not really on the screen. I'm just kidding.
So the system originally, and forgive me for boring people that have already heard this multiple times, but originally we had tried to reach out to the commercial systems to kind of see what would work, or if it would work, and if we could customize it to fit our needs. And it didn't work in the beginning. So we did create our own system, knowing that at some point there are commercial systems out there and that there will be some need for integration, crosstalk between the systems. So the intent is to have that built, but that's not going to be available on day one. And that will be, I think we'll probably be actively working on that in the 6 months between 6 or 8 months between when we release it and when it goes to a required action.
Okay. Any questions from the in-person attendees here regarding that? Okay, so I guess, of course, from my perspective, chairing the Pharmaceutical Science Committee at SWOG, my first priority is to make sure everybody at SWOG is well cared for in our best ability. So as I have been learning about the system as well as what we currently do, I would just like to share some of my thoughts. I do think that we have to be very mindful of when you're using a dual system. Oftentimes there will be duplication, and so anytime when we duplicate things, that you increase the potential risk of error. And so consequence to that, I think we also need to think beyond just the immediate day-to-day thing to think about audit. So we are already discussing that at the SWOG Quality Initiative Committee, which composed of representation of all aspects of protocol development and execution also. So we will be following this very closely and to learn about what, and even among different sites, there are a very diverse level of best practice as well. So, part of my plan is to try to learn as much as I can from what people are doing at various types of investigator site, and then to see if we can come up with some recommendations so that I think in this case, my personal feeling is that the transparency and the open communication is critical to make sure that we don't create additional work for ourselves in all different departments also. I mean, I completely understand why NCI want to develop and roll out Aurora. But the same token, what we are hoping is to really get them start talking to each other and start dating maybe, when then we'll see, and see if we can really create something that it has less of a duplication and allow them to integrate better. So while I'm talking, there's a second question is, do you anticipate that the timeline of integration will occur before the mandated use of Aurora EDARs?
That's a good question. I don't know. I think that it's to your point of speaking of the collaboration, the open communication. I think it's very important that we have productive discussions and not confrontational discussions about how we can move this forward, because Aurora and the EDARF is coming, period. Right. So it's a matter of how we can make it work and how we need to get together and how to have a constructive conversation about moving that forward. Now, as far as whether it's going to be available, those types of integrations are going to be available prior to the final implementation, that's going to be dependent on, we obviously would love to have feedback from sites that is constructive and that might help us help you or help you help us. But that obviously will also depend on the collaboration between the commercial vendors.
Yes.
That's something that's going to have to have there's going to
Mhm.
have to be a conversation
Right.
between the NCI and the commercial vendors. And, we may need some help facilitating that with
Mhm.
people that are, we hear pockets here and pockets there of people. Well, I talked to my vendor and they said that they didn't hear anything about this, or they don't know anything about it. And then, we need to get the right people in the room
Right.
to have the right conversation. And,
Mhm.
we built a system with the ports to be able to transmit information and to transmit things back and forth. So it's a matter of how we can make it work as one, as a federal system, and two, as something that is not, we're not trying to compete with the commercial vendors.
Right.
It's a matter of what we need to do to be compliant with our sponsor responsibilities.
And NCI is open to working with the vendor, but there are certain restrictions because it's a federal agency,
Correct.
right? So I think that is a very important aspect to recognize that the want to work, the want to integrate is there, but perhaps we, all of us, at different level in the, I call it the real time, to really think about perhaps many of us who are in the practice side, you have a connection with the vendor, maybe you have contact with the vendor, talk to them and see their response and really encourage them for that. And at the same token, we as in SWOG may think about how we can help to champion that relationship also. It's just a win-win situation if we can get that happen. But we need to work on it, and I think there's a good opportunity for that. We just have to push forward.
Yeah, agreed. I think and I know that there have been people that have reached out and said, well, the person that works at our site, the vendor that we work with, is willing to talk to your people, but I can tell you that we're not quite ready to do that yet. We're trying to get the system stabilized on our side and up and running. So once we hit that January release date, I think that the conversation changes, and then we can start doing some engagement with more deeper engagement with sites, deeper engagement with the vendors.
Right. And it seems like the impact of the integration is more with the phase 2 instead versus the phase 1 also,
right. I mean, nothing really should change. It's, ordering.
Okay. That's good because it helps to have a certain timeline. Then we can start planning and start talking also for that.
Yeah, and people will get a feeling. I don't know if who in the room or online saw the OECU presentation, but you'll have a feel for the system. It's not, when we say Aurora, people freak out and think that it's only the EDARF. There's a lot of other things that go into the system, and
the dashboard you show at your presentation was very helpful.
Yeah. So I'll take this opportunity, if I can take a second, to plug the training materials. This is obviously a massive rollout to thousands of sites and tens of thousands of people that are going to have access to the system and have to pull information from the system. So we're trying to do our best to create a resource page on the CTEP website. So you're welcome to check that out. You can ask questions through there. We have an FAQ document. We have training videos similar to what we had for the last system. So you can get a jump on that and get a feel for what's coming. Most of that is from the EDARF perspective, but there are overview presentations that are 3 to 5 minutes that you can pop in and pop out of. So I would recommend checking there before you start blasting questions.
So I understand that with the phase 2 rollout, more people will need to be registered, right? Is this called RCR? I always cannot get all those alphabets.
So there's different levels of registration that are going to be required. It depends somewhat on your site requirements, and if there's a group level requirement that you have to have GCP or other advanced training to register at a certain level. But if you're an AP, an NP IVR, or an investigator IVR, those would go through RCR, but you will be able to access it. Most of the people I would say probably a majority of the people that are ordering designees right now are associates, so you'll still have access at that point. But if, with the phase 2, anyone that is going to need to document on the DARF is going to need to be registered at least at the associate level,
Associate level.
which is the IAM level.
Right. And how extensive is that registration? Can you give an approximation of the time commitment?
The time commitment for actually putting
And
in the
don't
IAM.
include me in that. I need special care.
The IAM information that we collect is pretty minimal. We have implemented the ID.me into that identification process into that. So that takes a little bit of time, just collecting documents and getting the authorization and getting your verification. And then that also includes the 2-factor authentication, which is going to be required to get into the system. So part of this digital identity guidelines that require the ID.me authentication and multi-factor. In addition to that is documenting who has access to the system, which is one of the reasons why we have to have people register. So as a federal system, we need to be able to answer that question, who has access to our system.
Does it need to be updated annually?
So the IAM and the RCR are a little different. The IAM was, you had to re-register annually. The RCR, you have to, IAM, you had to redo your password every year. RCR, you have to redo your password. It's every
More often,
120.
yeah.
It's more often.
Yeah.
But with that's the good thing about ID.me. Once you have your ID.me account set up, you never have to change your password ever unless you want to, and that's through ID.me. So we're no longer with the IAM process managing the password because we have a link to ID.me. So that's a benefit of transitioning to ID.me for sure.
But the RCL you do have to update, particularly if you have the certifications, right?
So you have, so that's two different things. If you're talking about access, that's the access is through your IAM credentials or your ID.me credentials.
Okay.
But to keep a current active status within RCR, you do have to register on an annual basis.
Okay. So obviously this potentially can impact many people in each of the institution. For the phase 2, would you recommend people to start working on that at this point? Just to think ahead.
Sure. I think that you can start thinking about who's going to be involved in your research and who's going to be involved in ordering drug, who's going to be involved in documenting on a DARF. And those people might be the same people that you have now,
Right.
that are doing the work, and they may already be in your PSD package. But if you think that you're going to need to expand that list, then certainly you can start anytime.
Okay. All right. I don't think we have additional questions virtually. Nope, I think we addressed all of them. And is there any additional questions or concerns in person in the audience? All right. As we move forward, Matt, I'll keep you updated also with the discussion within SWOG as well. Thank you.
Okay, great.
Okay, wrong button. So, Andrew.
Thank you. So just have very brief updates. I have a few metrics on the next slide.
Yes.
So the past year has been somewhat busy since the last meeting. We've had 17 new drug requests. So you can see the drug name, the corresponding committee, and those who wrote the section, as well as the reviewer. And then the next slide has the 15 updated drug sections that we completed in the past year. So 32 requests overall. The next slide, please. Just wanted to also take a moment to thank all of our writers. And as Sufan said, we are in our 3rd year of utilizing the learners in our training programs. So this year we have who's the PGY2 investigational drug and research resident at Johns Hopkins, Elaine, who's the Chapman Amgen Oncology postdoctoral fellow, and then Lauren, who's the PGY2 investigational drug resident at Michigan. I can speak from the Michigan program. It's a very valuable aspect of our training program, and I think it provides a lot of good feedback and training to our residents, and it's a valuable experience. Just want to again thank them for their time and effort. And then Caitlin Young and Michelle Yu, who are in the previous years, have remained engaged and continue to write and review sections. So I want to thank them for their time, as well as all the reviewers. I was very appreciative. I send emails occasionally and nobody ever says no. So I appreciate everybody leaning in and providing that feedback to the writers and being able to help write all of those sections. So that's all the prepared slides. I just had one additional thought that I did not include. We've been asked to help write the wallet card and patient handouts for certain agents. So we'll be following the guidance in the NCI protocol template for which agents need that wallet card. I think it's a small subset, primarily the non-FDA approved drugs that have drug interactions. So we haven't crossed that bridge just yet, but when we do, I'll be in touch with the writer and reviewer. We can walk through that process together, but it shouldn't be a large additional work, just kind of formatting taking the interaction information that we already wrote and just turning it into a patient-friendly language. So that's all that I have.
Okay. And then I think the last item on the Drug Information Subcommittee is, oh, sorry, yes.
So Andrew, so the drug wallet card handouts, are you talking about agents that are not CTEP agents? Or are you talking about just in general when you get a new agent?
We would follow the guidance in the table in the protocol template. So I can't quote it exactly, but I think it's the non-FDA approved CTEP supplied agents that require the wallet card.
Okay, so we do generate those if they, we have the drug interaction task force that reviews all of the drug information that we produce, and then we create that and then they review it. So there are wallet cards already available for a majority of the agents.
Great.
So if you do come across one, feel free to shoot me an email or give me a call and we can let you know if we have it.
Great. Thank you so much.
Okay, the last items on the Drug Information Subcommittee has to do with the protocol review, because we do get requests to review protocols also. So while I received a message from Holly, who in charge, Holly Chan from City of Hope. She's in charge for the heme side, and then we have another member doing the solid tumor. I basically receive information from them is they have not been receiving any protocol review requests, which is a little concerning. However, on the side, I think both Andrew and I, in addition to managing the Section 3, we have continued getting a lot of questions either on active protocols as well as protocol in development. And I personally actually work on a very complicated lymphoma protocol in terms of the whole protocol development and rewrote some of the sections also. So we continue to provide a supportive role in that sense. But I do get concerned that when the kind of protocol already ready to be submitted, we are not being asked to review, because oftentimes that produce some important feedback to the investigator. So I guess I will look into it. Crystal, maybe I can talk to you about that and see if maybe that step is not occurring, or we'll see. Yeah, but I would like to continue that, because I think we created a whole very structured system to accommodate that. And I do feel that the review, particularly with the complexity of many of the protocols and so many oral agents being used, I would like to be more engaged, with more pharmacists engaged in that process of that. But anyhow, I think that completes the section, and then now we get to item 3 on our agenda. Is a special presentation by Dan Hurst, our PharmD PhD from University of Michigan and the faculty at the School of Pharmacy also, and a translational scientist. So he's going to talk about opportunities and challenges as a translational symptom scientist as well. Thank you, Dan.
Thank you. Can I have the slide advancer?
Sufan?
Huh?
Oh, yeah,
sorry.
that's okay.
I thought you were talking to
No. Cool. Okay. Hi, everybody. I'm Dan Hertz. So I was asked to give this presentation yesterday to the Symptom Control and Quality of Life Committee to give them a sense of the kinds of translational sciences we do in symptom science and toxicity. And Sufan asked if I could come and give a similar talk to you guys. Because the committees are a little bit different, I'm going to focus on slightly different aspects and talk more about kind of the process and how I think if you guys are interested in sort of clin pharm or translational medicine or research or biomarkers or anything, how maybe you could go about getting involved in doing some studies in SWOG. I'll spend a little bit less time on the toxicity and on the actual data itself, because I don't think that's really necessarily of interest to you guys. Okay. So here will be the outline. I'll just start by telling you a little bit about taxane-induced peripheral neuropathy, just so everyone has sort of a baseline understanding of the toxicity we're talking about. And then I'll go relatively quickly through two projects that kind of illustrate the two different ways to do translational sciences research within SWOG. And then I will just finish up by making a couple general comments about the current process and where I think we're going in toxicity translational science. So most of you are probably pretty familiar with taxane-induced peripheral neuropathy. It's a common side effect of the taxanes and several other classes of chemotherapy agents, and it occurs in the fingers and toes and starts with like a numbness and tingling and can lead to loss of function. There are currently no effective strategies for prevention or treatment, and we also don't have any clinically useful predictors of peripheral neuropathy. So all we can really do is if a patient is experiencing neuropathy, we can adjust dosing in some way, or sometimes we have to actually discontinue treatment, which can reduce treatment effectiveness. So it's one of the more clinically relevant toxicities that people are really interested in finding predictive biomarkers and treatment strategies for. Most of you are probably familiar with the NCI CTC AE scale. These are the definitions for grade 2 and 3 peripheral neuropathy when it's assessed by clinicians. A lot of toxicity in general and CIPN specifically are moving towards patient-reported outcome scales, which are considered to be more sensitive and reliable and tend to be preferred for these studies, even though I have actual concerns about using them. Okay, so before coming to SWOG with an idea that you want to get samples and data and do some analyses, you're going to need some kind of preliminary data because you are not really supposed to be doing discovery phase science in SWOG samples. You're supposed to be using SWOG samples to do sort of validation studies. So here is the clinical study that we got our pilot data from. This was a single site observational study of 60 patients who had early stage breast cancer and were starting standard of care weekly paclitaxel. We collected the CIPN20 patient-reported neuropathy instrument at baseline and weekly throughout treatment. And you can see that on the bottom right side. On the x-axis is just weeks of treatment. On the y-axis is the severity of neuropathy. So a lot of variability, but over time an increase in neuropathy as you'd expect. So in addition to collecting the PRO phenotype data, we collected lots of different biomarkers, including samples, so we could look at vitamins. So I'll tell you a little bit about our vitamin D data, and samples. So we could do metabolomics, which is a science where you measure sort of endogenous molecules, and you can look at dynamic changes during treatment and how they might predict response. So cutting right to the results of the pilot study, we saw that patients who had vitamin D insufficiency prior to treatment had higher risk of neuropathy. So in this figure on the y-axis is the severity of neuropathy, and this is stratified on the left, patients who are vitamin D insufficient, on the right, patients who are vitamin D sufficient. So again, this is sort of a discovery phase study. There are other similar retrospective analyses that have found similar kinds of things. So this was pretty interesting to us. We also saw, again, a very biomarker discovery phase result that patients who had low histidine concentrations seemed to have higher rates of neuropathy. And we thought that this would be worth following up in a much larger patient cohort. So that's what brought me to SWOG. I was already involved in SWOG, but it brought me to this SWOG 0221 clinical trial so we could try to validate these biomarkers. So SWOG 0221 was a prospective randomized controlled trial where patients were randomized to two different schedules of ACT. So there was 2 schedules of the AC, 2 schedules of the T. They found out that it was pretty similar in either schedule. But importantly for us, this study collected CIPN data both on CTC-AE and on the patient-reported outcome in a subset of patients. And most importantly, this study collected and banked serum at baseline. So this was sort of the perfect opportunity for us to try to validate some of our biomarkers. There has been prior work done in a subset of the patients from 0221 showing that patients who were taking multivitamins had some neuropathy protection, and then some dietary work showing that patients who had higher grain intake were also protected. So that was interesting to us because we saw this vitamin D association, thinking, maybe it's the multivitamin delivering vitamin D and that's why they're protected. So if we can get our hands on those samples, then we can sort of mechanistically validate that. So that was our objective for our translational medicine proposal. And then skipping directly to the results, we found, as we hypothesized, and as we had seen in our pilot study, that patients who were vitamin D insufficient had higher risk of grade 3 or higher CIPN. So that's the figure on the right-hand side, again, stratified by insufficient versus sufficient. I'll probably skip the results here. You can see that about 15% of the patients had CIPN and about a third had vitamin D insufficiency. So this is not like a rare phenomenon. And right now we're not measuring it and we're not supplementing. So I think there's been some discussion based on these results, given that this is sort of we call this a prospective retrospective study. So a retrospective analysis of a prospective clinical trial. This is like in biomarker sciences, how you validate a biomarker. So now that we have this as maybe a validated biomarker of CIPN risk, the question is, should we just be supplementing vitamin D? There was a robust discussion in symptom control and quality of life yesterday about sort of the concerns with doing that and how we would actually have to go about prospectively demonstrating a clinical benefit in doing so. But I think that some people might take this information and say it might be worth trying sort of vitamin D supplementation. After we got those results, we reached out to a group that does some mouse neuropathy work. They have sort of an established mouse model of taxane-induced peripheral neuropathy. And we asked them if they've ever done any work in vitamin D deficiency, and they hadn't, but they found another paper where there's an established model for vitamin D deficiency, where you just give mice, they start out normal, you give half of them a vitamin D deficient diet for 8 weeks, you give the other half a normal diet. And you can see after 8 weeks of just the diet, here are the mice with the vitamin D deficiency diet. So they have a pretty robust change in mechanical threshold sensitivity, which is like a mouse phenotype of neuropathy. So without even treating them with paclitaxel, you can see just inducing vitamin D deficiency causes them to have a neuropathy-like phenotype. So mechanistic evidence that this might be a causal relationship, but again, we still probably need to do a prospective clinical trial. The histidine was not validated. Here's sort of box plots looking at the patients with and without neuropathy, and you can see complete overlaps. So that was a false positive in our initial discovery study, as is pretty common in biomarker sciences. Okay, so if you were interested in doing some translational medicine in SWOG, here's sort of the process that you need to be ready to put yourself through. So we presented our preliminary data to SWOG in 2018. There's a lot of excitement. This was done through the Breast Translational Medicine Committee. It took us almost a year to go through the process to get SWOG translational medicine approval. That's partially because we did the process kind of backwards. That process itself probably takes about 4 months, 5 months. If you're working on this and moving it forward as fast as you can, you need to be really persistent and you need to be really assertive to get anything through these processes. There's lots of rounds of review, lots of people who want to look at it and give you feedback and things. So several months to get just this internal SWOG translational medicine approval from the time that you initially propose it to a committee. And you have to sort of start with that. Okay. So after we got SWOG approval, now we have to go to NCI Navigator. So that's the NCI system that has control over all the samples from SWOG and Alliance and ECOG. So any of the samples that have been collected, you need to go through Navigator and get their approval to get those samples to do any work. Once you have that approval, which takes another set of months, you then need to get funding, right? So we started sort of concurrently trying to get funding, several proposals to different groups, ACS, NIH, SWOG, et cetera. That took a little while. On our third submission to the ACS, we finally got a fundable score. So we were 3 years into this project and we hadn't started doing any research, right? So like that's the process that's sort of how it goes here, right? So if you want to get your hands on samples and data to validate a biomarker, you're looking at 3 years before you start doing any actual science. Once we got the funding, we got the samples, we started doing the analyses and all the data that I showed you today. So that's taken us, I think, less than a year. So the research isn't what takes a long time. It's getting the approvals, getting the funding, getting through the process. So I'll skip through that. So that's if you're lucky enough to have a study where there are samples and data banked and collected and available for you to do your research. The other option is that study doesn't exist and you have to actually run a prospective study to get the data and samples. So that's what we did in SWOG 1714. So in addition to the vitamins and metabolites in our pilot study, we did several other types of translational medicine biomarker sciences, including pharmacokinetics. So we collected a sample right at the end of infusion to look at a Cmax. I don't need to explain that. So the Cmax to the audience in this room, you guys are pretty familiar with PK. We also looked at pharmacogenetics. That's sort of my background. We were interested in genes that have been associated with hereditary neuropathy conditions that seem to often pop up in genome-wide association studies of taxane-induced neuropathy. So again, skipping right to the results. Here are the results of the PK analysis. So we divided our cohort into tertiles of Cmax. You can see the patients in the highest tertile are the ones that had the most dramatic increase in neuropathy over the course of treatment, whereas the patients with the lowest Cmax had a lower increase or lesser increase in peripheral neuropathy over the course of treatment. This association with PK has been repeatedly demonstrated. I would say by far the most important predictor of taxane-induced or paclitaxel-induced peripheral neuropathy is the paclitaxel systemic exposure, as measured by either a Cmax or like a pseudo AUC. So we just sort of confirmed that in our study. We also saw this interesting, although very discovery phase, again, association where patients who had this variant in EPHA5 that has pretty consistently popped out of the genome-wide association studies and been replicated seem to have higher risk of peripheral neuropathy if you adjust for sort of cumulative drug administration and systemic exposure, right? So I think the reason that we're not finding a lot in our pharmacogenetic studies is because we can't account for PK. In this study, we measured PK, so we could account for it, but that's what it is. Okay, so SWOG 0221 didn't have PK data, didn't have lots of samples, didn't have the best phenotype data, and there was an interest from Dawn Hirschman and some of the other people in the sort of symptom science world to do a prospective observational study of taxane-induced peripheral neuropathy so we could try to find predictors, including clinical variables and also biomarkers. So that's this study, SWOG 1714. The primary endpoint in this study is a change in the patient-reported outcome scale. So that will be the same endpoint that we use for our analyses. So my involvement was mostly in developing secondary objectives in terms of translational medicine and correlative biomarkers. So here's the schema for that study. It's very similar to my study. It's an observational prospective cohort where patients who are starting standard of care paclitaxel or docetaxel are enrolled. In this case, this study is much larger. 1,350 patients, and they're followed for much longer, so out to 3 years. And in addition to doing the PROs, we're doing sort of a battery of toxicity or neuropathy phenotype assessments. So objective measures, patient-reported measures, clinician assessment, all sorts of things that we think might be indicative of how much neuropathy these patients are experiencing, so we can do our best sort of predictive biomarker work. So we have built in lots of sample collections so we could try to validate some of our predictive biomarkers. So we actually are doing PK. It's a mandatory PK sample, again, right at the end of infusion in every patient, just the first infusion. Also looking at pharmacogenetics, so we can try to replicate this EPHA5 or do additional discovery, vitamin D, metabolomics, lipidomics, basically anything we can do with a serum or plasma sample are all sort of in the plans. So I told you about the timeline. If there is an existing study with samples and data, here's the timeline if you want to start your own study to collect your own samples and data. So as far as I can tell, this was initially proposed as a concept in 2016 by Dawn Hirschman. It was disapproved because there wasn't interest or whatever it was. A little bit after that, the NCI actually had a CIPN clinical trials planning meeting where they brought a lot of experts together and said, what do we need to do to move forward on CIPN because we aren't making any progress? And there were sort of two recommendations from that. One was to start treatment trials for a specific class of agents that we thought might be helpful for treating neuropathy. And the other was to do a sort of observational study where we can collect samples and data to do biomarker work. So essentially we proposed back to them Dawn Hirschman's study that they had previously disapproved. And at that point, they thought it was a great idea. And that's kind of when I got involved as the translational medicine chair. So that was sometime in 2017. It took a little while for it to go through the whole SWOG development process. It was activated in 2019, so now we're almost 3 years into the process. Concurrent with that, we started submitting translational medicine proposals so we could do whatever translational science analyses we want to do. It took 3 separate proposals for reasons that aren't worth going into. This study actually accrued very robustly, even despite COVID, and accrual was complete in late 2021. We completed about 1,350 patients, so we're 5 years into the project. Soon thereafter, we submitted an R01 to do all the translational medicine work, right? So we collected all the samples on the study that was paid for by SWOG, but there was no money in the protocol to actually analyze the samples. So we wrote an R01 to do the translational sciences. That received a very strong score in the 6th percentile. We got that news, I think, in August-ish. We are expecting a notice of award any day. We haven't received anything final, so we are not ready to celebrate yet, but we think based on that score that most likely we'll be able to move forward with this work. But if you look at the entire timeline, we are 6.5 years into this project and we haven't started doing any translational medicine research. So here are the aims from the proposal. The first aim was to look at sort of physiological biomarkers. The second aim was to look at genetic biomarkers. The third aim is to integrate whatever biomarkers we find into the clinical risk prediction model that is being developed as the primary objective of SWOG 1714. So just some comments on SWOG translational symptom science. So as I've mentioned, there are sort of 2 options for doing translational medicine studies within SWOG. One of them is using existing data and samples. It's a slow process with lots of round of review. The second option is prospectively collecting your data and samples. It's an even slower process with even more rounds of review. Either way, these take a long time, but if you want to be doing practice-changing biomarker research, you need to go through these processes and you need to do it in this context. It does help a lot if you actually know the process and work with the SWOG staff. There's always this issue about whether you should try to get funding first or try to get approvals first. I recommend getting the approvals first, and those approvals will be contingent upon you getting funding. So when you get NCI Navigator approval, they say this is approved. You need to demonstrate to us within 1 year that you obtained the funding to do this work. Otherwise, the approval will be vacated or void or something. You can extend those, but that's sort of the process. You get the approval first, and then you can submit your R01 and say, I have approval to get these samples. It's contingent upon this funding. The last thing is that I think I've tried to make it pretty clear that this is a long process with lots of review that requires real expertise. So I do not recommend just like clinical investigators try to do their own translational medicine without bringing on board methodological expertise, statistical expertise, all the people that actually know and think about deeply these fields. And that expertise does exist within and outside of SWOG. So if you are interested in doing this and you think, oh, we should really do a pharmacogenetics analysis of this study, there are pharmacogenetics expertise on both the methodological and statistical side that can help you. So I would strongly recommend bringing that expertise. I was also asked to comment on the future of translational symptom science. I'll go through this quickly. I think the future is advances in how we collect toxicity phenotypes, so using PROs, using wearables, objective measures, all those kinds of things. Advances in the science that we can do on the methodological side, so single cell analyses, microbiomes, all these kinds of like futuristic science things that we can do to collect more data from different kinds of approaches. And then there's been huge advances. If you were at the translational medicine plenary yesterday, you heard a little bit about AI and machine learning. So we are using a lot of AI and machine learning to do complex biomarker analyses, especially integrating across biomarker classes. The challenges is that a lot of times when you're starting to think about new approaches, you get a lot of very discovery phase things that don't replicate and validate in the future, that are considered to be false positives. And a lot of things that we'll get from these signatures, if we make complex signatures that include kinetics and genetics and lipidomics, they're going to be very hard to interpret. You'll have these sort of machine learning algorithms that'll tell you this patient's at high risk or not high risk, and you won't really know what is causing that and how to intervene. So thinking about how we actually translate these into clinical practice is pretty complex. So I think that it's going to be very challenging to translate complex signatures for toxicity prediction into clinical practice. And I think that that complexity is something we should think a lot about, particularly in the context of the current situation where we have very simple, very strong predictors of toxicity. I'm referring specifically to DPYD genotyping to predict fluoropyrimidine chemotherapy. I never miss an opportunity to tell people about this. It is standard of care in Europe to test people for DPYD prior to fluoropyrimidine chemotherapy. In the US, it's not, and it's not used very much. This is a very simple, very cheap, highly predictive test that can tell you whether or not a patient's at risk of severe, potentially fatal toxicity, and we don't use it. And clinicians don't use it, and the clinicians in Symptom Control and Quality of Life Committee, a lot of them don't use it. So if we're not going to be using very simple, very strongly predictive biomarkers, then I hate to say we shouldn't fund and waste our time conducting further analyses to find more complex biomarkers, because they're not going to be as simple and they're not going to be as predictive as this one that we already know about that we are currently not using. So that's my sort of pitch for everyone, to get your clinicians to use this, the pharmacists. We have been working a lot with the pharmacy societies, HOPA, ISMP, different people, to advocate for and endorse pretreatment DPYD testing. If you are working with clinicians and they are not testing for DPYD, you should tell them that they should. I'm hoping that maybe at some point pharmacists will say, I don't really think it's safe to administer this drug or fill this prescription without a DPYD test, because these are avoidable toxicities and deaths that are occurring daily. Thank you very much. I'd be happy to answer any questions.
So I'll start. No, don't go.
Oh,
away
okay.
yet.
Oh, I'll answer questions all day. I love talking about my research.
So why do you think the clinicians are not using the biomarker? I mean, UGT1A1 is a fine example, right? It was like the first one that get on the package insert. But I think everybody here would agree with me that hardly anybody used the UGT1A1 as a pre-administration testing. Also, I hear reasons, anecdotally, but I'm just curious.
So I think UGT1A1, there's less clear clinical utility of that, because you are preventing toxicities that are pretty manageable and are otherwise resolvable. For the DPYD, there's very clear evidence of the clinical benefit. So there's clear evidence that using this prospectively reduces severe toxicity. It actually reduces healthcare costs. When you talk to the clinicians, we actually did a survey through SWOG asking clinicians whether they test, and if they don't, why not? They said the number one reason was that they perceive this to be an uncommon variant or an uncommon concern. And the second issue is that the NCCN or ASCO guidelines don't tell them to test. So really, clinicians are waiting on the guidelines to recommend testing. We have submitted requests to the NCCN guidelines multiple times. We actually had, we had submitted something to the Institute for Safe Medication Practices where they endorsed testing, and they asked the NCCN guidelines co-chairs for a comment. And their primary concern is that if you reduce dosing, it might reduce treatment effectiveness. So that article that I just pointed out that I published in JCO goes through all the evidence. There's no direct evidence of that. And there is evidence from PK studies that the recommended dose modifications normalize systemic exposure. So there should be no reason to believe that efficacy will be reduced. And also the evidence from the studies that have been done is that those recommended doses are right at the maximum tolerated dose. So we think that those are the appropriate doses to use, but the NCCN co-chairs have been pretty adamant that they want to see prospective evidence that patients who are variant carriers and received reduced doses do not have any reduction in efficacy. We asked them how we would possibly do that study, and they said they don't think it's possible, but they're hoping that maybe there will be real-world evidence from the implementation in Europe. Or they said they would accept real-world evidence, and I think that we will get that from Europe, but it'll take a couple more years, and we will unnecessarily kill a few more patients, or a couple thousand more patients.
So do you see that's parallel to what we are seeing with this whole field of use of pharmacogenomics data? I mean, people are getting all this genomic testing done, right? Patients even, but rarely do we see people actually use the results and apply that to care. But do you think it's because of that we need to bring up the education level?
Yeah, I think there are sort of a lot of things going on. So one is that across pharmacogenetics, we found relatively few things that are actually clinically useful and should be used preemptively. So like I think clopidogrel is actually clinically actionable and should be used in practice. Now we have prospective randomized trials demonstrating that, but they're still not using it for clopidogrel. Abacavir is very clinically useful, and now it's sort of required that you test for HLA-B star 5701 before administering abacavir. So that one is in clinical practice and is used commonly. I think that generally there's just somewhat less interest in toxicity than there is in efficacy. So it is not lost on me that the tumor genotyping that determines treatment effectiveness has been sort of widely adopted in clinical practice on sort of similar strengths of evidence as we have for DPYD. So I think that's just an issue of toxicity versus efficacy. Yeah, there's sort of a lot into it. I think in pharmacogenetics, we talk a lot about like the lack of education, the challenges with implementation, how do you pay for testing, the reimbursement, and all those things. And I think those are challenges, but I think those will all be surmounted once the medical oncologists actually want to do this test and are accepting of it, because all those same challenges apply to somatic tumor testing and have been, overcome. And I think if we can do it there, we can do the same thing in pharmacogenetics.
So I have a question that's not science, but so I would ask, like, based on what you present with the timeline and seemingly discouraging to you, but you're still doing that.
It's not discouraging to me, and I'm not trying to discourage anyone else from doing it. I just think I think you need to really decide that this is something that you want to be putting the next 5 to 10 years into, and this is something that you think might actually lead to clinically useful practice-changing findings, because otherwise it's just not worth the amount of time and effort. So like you really need to be committed to doing this. I wouldn't necessarily recommend doing this as an assistant professor because you have a timeline and it might not exactly line up with the timeline that is required to get these studies done. But I think that if you have a good idea and you have preliminary data and you want to move a biomarker into clinical practice, you have to do it through this process. Yeah. You can do all the sort of retrospective small replication studies you want, and no one's going to pay any attention until you do it in a prospective clinical trial, and then actually get one of these committees to then take that into a prospective clinical trial. So we're talking to the Symptom Control and Quality of Life Committee about a vitamin D prospective clinical trial. We're talking to that same committee about a prospective DPYD clinical trial. They're challenging to develop. There's a lot of resistance to sort of biomarkers in pharmacogenetics, but again, if you want to actually affect patient care from a sort of biomarker perspective, this is the way to go about it. And I'm very excited about it. And I'm hoping that more people get more involved and we can do better science, because there's a ton of data and a ton of samples, and there's just a very few a very small group of people that have this sort of clinical pharmacology and biomarker expertise to actually run these kinds of studies.
Yeah, and I also agree with you. I mean, finding a good mentor and as well as really learning the system helps a lot. I mean, I watch it on the other end as I'm seeing people submitting capsules and things like that. I can see that. So really part of that is the non-science part is really understanding the system also.
Right.
Yeah.
And thankfully, SWOG has great staff members that know what the process is and where the forms are, and they are extremely helpful and really invaluable in this process. So my thanks to all of them that have helped along the way.
Okay.
Thank you.
Thank you, Dan. Can you switch back to the meeting slides? Thank you. Oh, okay. There we go. Thank you. So now we're moving on for the remainder of the agenda, and I'm trying to pull something up here myself. All right, so Jane Rogers from the GI committee and also a PSC committee also member also has been working on a Beacon standardized order set updates. I briefly touched base with her, and there's no update, so we're going to table it for this meeting, and we will look forward for her continuing report in future meetings. Dan, again, is anything on digital engagement committee?
Yeah, I mentioned earlier the drug interactions and smart forms project. Digital engagement right now is undergoing a change in leadership. I'm not sure how much of this has been announced, but the current leader is taking over a leadership of a different, right, the DEI committee.
Yeah.
So I think we're currently looking for new leadership within digital engagement. A lot of things that have been done in digital engagement focus more on like the use of social media to increase awareness of studies and sort of some of the things that were talked about during the plenary yesterday. How do we use digital tools to increase enrollment and awareness of clinical trials? There's been less done on this sort of wearables and other like actual science aspects of digital engagement. So I'm hoping that we bring in new leadership that wants to focus more there. I think that would be perhaps more relevant to some of the things that we think about in this committee.
Okay, thank you. All right, so the next agenda item has to do with pharmacist liaison report, and usually we review our liaison assignment list. This is the current assignment list, and typically we like to have 2 liaison for each committee, and you can see that we are doing pretty well, except in GI. We only have Jane on as the sole liaison. So if anybody is interested with the GI, or you're also welcome to switch if GI is your higher priority interest also. Just shoot me an email, and then I can work with that. Typically, the way that I do that is I ask each individual to rank their priority of their desire, and then I try to do the matching in that way. So we're doing pretty good right now. Actually, this is the most field I seen for a few years. So if anybody is interested in the GI committee, please do let me know, and then we can work with you on that. Yes, Crystal.
It's about 2 years or more outdated on the protocol and committee leaders. So
Yeah, that has been switching, and this was sent to me most recent. Yeah, so we help me.
We updated it previously, but
Okay.
it's a little outdated.
Okay. Yeah. So I will work with you to update those. Yes. And are we pretty good now with protocol coordinators in terms of?
That's a good question.
Okay.
We're going through some changes right now. We've promoted a couple protocol coordinators to project managers. So there's definitely been quite a few ongoing changes in the last month,
Yeah.
this week, and
Okay, all right. So with that, the next item is just an open agenda for any members of Pharmacist Liaison to raise any questions, or if there's any interest in certain areas and or what they learned from their respective disease committee, to bring it up for discussion. So it's an open discussion. Oh, that's right. So I am just watching the chat and see if anything come up. Any one of you has? Okay.
I just want to share one of my experience, kind of unique, I think, when writing the drug section. So SWOG 1800D, the study agent is N803. It's very unique that the information that we got from the package or from the sponsor or manufacturer, it says that there's no stability information. So the instruction for preparation is to prepare it and then immediately give it to patients. So that's kind of how it, I think I didn't write a section, but actually I was on the more like the dispensing pharmacy side when we saw that instruction. So it creates some challenges and question and required the site to kind of ask for further clarification, because as some of us that as a practitioner, we know that it's kind of not feasible to be able to prepare something and give to patient immediately. I believe most of the sites that will require pharmacy to prepare the agent, especially in oncology, we would not like allow nursing to just drop at the bedside and give it right away. So it create some confusion and challenges. So we have to reach out to really like try to, ask what is the minimum or maximum time that, allow us to prepare or the stability, basically, we want to ask. We did not really get a clear answer still, but we told them that that is the fastest that we can do. Like, whatever, I forgot what we put internally, like 15 minutes or something, to say if it's acceptable. So I think the lesson I learned is kind of remind myself when we are doing the drug sections, writing those instructions, to be more mindful, and if certain things that we as the committee member writing the sections can get some clarification prior to implement in the protocol. I think that will help each site saving their time and I think helping a lot. So I think that's kind of lesson I learned, and I kind of try to remind myself when we're doing those drug sections to kind of pay more attention and things like that. But I think it's a very unique, and this is the first time I've seen. Usually we got a lot of good information from either NCI or the sponsor or not sponsor, the manufacturer. So
do you recall, is it a CTAP drug or is it
I don't remember.
industry?
Do you know if that? Oh, it's a SWOG IND. So it's the industry can provided the IB and information.
Yeah, so with 19D, or 18D, sorry, we did know in development that this was going to be an issue. We've had many conversations with pharma and trying to kind of extract that information from them. And we are literally waiting on that stability data. I think we're supposed to have it October, November timeframe. But it was a concern during development, and we pointed it out, and it's still a concern. And it could be actually some of the problems that we are seeing and accrual to that study because of the logistical obstacles. And there's a few other items there as well, but we're definitely aware of it. And we've been pushing the company as much as we can to get that information for us.
I know we have been working from our side also, trying to get them to release that information.
Yeah.
And I know it's been delayed a few times, so.
Yes. We're aware. We knew it was going to be a problem, so yeah.
But do we know what is actually happening at some of the investigative sites?
With that study in particular, since they're also having some issues with having Pembro being provided commercially rather than being provided by the company. So there's definitely some barriers that we are trying to find. We've got our accrual enhancement committee working on identifying those barriers. And so the items that we've tasked them with is to reach out to the sites and figure out why they're not opening up the study, even though they've got patients being registered to those sub-studies, and seeing if that is a barrier for them opening it.
Oh, no, I'm talking about, but there have been patient enrolled, right?
Yes, it's just been really slow.
But do we know how they are managing the N803 administration?
I don't know that level of detail. Unfortunately, I didn't go to the site coordinators meeting yesterday.
Because I don't recall getting any inquiry, right, on that, so.
For me, I try to reach out to the protocol coordinator.
Oh, yeah, but that's where we usually get the inquiry from, is from the op office, not directly from, usually not directly from the site, yeah.
Yeah, you guys helped wrote the question, and we've had the back and forth with the
Right.
pharma company quite a lot.
uh-huh.
So, I mean, if we can't get the answer from them,
I know. Okay, other than that, is there any other agenda item that people like to bring forth? If not, then we will conclude our committee meeting today, and then look forward to seeing you in San Francisco. San Francisco, right? The next one, yeah. I didn't put the date, just, yeah. I just say spring meeting in 2023. Have a good day.